husband has MND but primary sclerosis. We have no offspring, but I was interested in the pre-implantation that you talked about, and I didn't quite understand it.
Right.
Not that we're going to benefit from any IVF of that. But are you implying that the gene is put into the mother-to-be so that the baby would not get, to create like a immunity or an antibody not to get the gene, or could the baby be at least years to come of getting the mutation?
So I guess this is a question around what is the basis of the pre-implantation genetic diagnosis at IVF. Well, this is offered to those families who know they carry a mutation within a family. One of those families that have SOD1 mutations or C9orf72, these are the major genes. There are a couple of others as well. For those families that know that the gene has passed down through their family. Now, we have family members who may be of childbearing age, typically too young to have obviously developed the disease, but they know they may have inherited that gene. They may not have been tested. They may choose to have that test or not, but they may have that gene. So now they want to ensure that they don't pass that same gene on to their child. And the pre-implantation genetic diagnosis at IVF is a means of testing the embryo, the fertilised embryo, to ensure that that fertilised embryo doesn't carry the mutation. So that's the embryo that gets transferred to the mother and hopefully leads to a successful pregnancy.
Is there any research related to stem cells for the sporadic MND?
Right, so from the stem cell perspective, I'll answer in two ways. There's certainly the interest in stem cell therapy. We don't do anything on that. But what we can do, we use stem cells as a means of mimicking the disease in the laboratory. It's really a very powerful research tool. So we can take a blood sample or a skin, we usually take a skin biopsy, just a small skin biopsy from a patient, and we can, what we call reprogram your cells to become motor neurons. So we can take a skin cell, reprogram it, and then we can grow a patient's motor neurons in the laboratory. And then that becomes a very powerful research tool for us to then look at the degenerating motor neuron and see if we can treat it. So it becomes a very powerful tool for drug discovery.
The Project Mine, is that right?
Right, right.
That is still ongoing, is that right?
Absolutely, yes.
Can you select your patients?
We still are. So there's a question about Project Mine and the Australian contribution to that international project. Yes, so we recruit our patients through at Macquarie, through the Macquarie Clinic, but also I showed you the slide around SALSA, which is the sporadic ALS, the Australian Sporadic ALS Consortium. And they are recruiting patients to research as well as controls, family members. And those patients are selected, particularly the sporadic patients, may be sequenced and contributed. It's a very expensive exercise. It costs around about $2,000 for us to sequence the genome. So we're talking millions of dollars for us to contribute, make our contribution. But yes, that is underway. Patients are recruited routinely and consented to research and consented to participate in our contribution to Project Mine.
Sorry, quite a bit of your PowerPoint was around personalised medicine. I'm just actually wondering, when you look at something like the trials in Therapeutic, have you been able to isolate where or in what type of group that particular drug would be most likely to respond?
Yeah, that's a good question. So the question is around personalised medicine, and can it be applied to clinical trials like the Tecfidera one that's coming up? Yes, absolutely. Those who are undertaking those trials will have a strong selection criteria in the first place. But being able to stratify patients, and it may well be done what we call post hoc, after the event, by going back and looking at the data and saying, which of these patients actually responded best? Was it a particular group of patients who had a particular genetic background? Did they have the particular genetic risk factors? That will be done. Impossible to do beforehand until you've done the trial, but they will be doing it retrospectively. And that's not just genetics too. There will be biochemical tests as well along the way. Also incorporate things like imaging and see whether they can. So there are multiple ways of doing this personalised medicine approach.
I did have a second question. We know that Lillytech is quite commonly prescribed in everyone who's been diagnosed. Have we gotten any, and it's the oldest drug available for treatment. So I'm wondering, there's been quite a lot of work now done in both projects. And is there any further progress to identify that genetic group that is most susceptible to the decline even whilst on the drug,
right,
and those that have got susceptibility of liver dysfunction?
So the question is around, has the same testing personalised medicine approach happened to clinical trials of riluzole? To my knowledge, no. And I think that's something that I imagine groups must be. The fact that it hasn't been published yet suggests that perhaps there hasn't been an obvious effect found, but that's a case of, have we identified enough of the genetic risk yet? We've only just started to identify the genetic risk factors. We've got 3 that I've illustrated today. There may be dozens, and we simply need to identify more, because it's not going to be one genetic risk factor. It's going to be 2, 3, or a combination of those. What are they and which combinations? And just with cancer, you'll have a series of tests that will you'll correlate and coalesce that data to identify which of the patients most likely. So we're tip of the iceberg. It's just starting to come through, but the UNC13A variant that I referred to, I'm aware of at least one study that is prospectively typing people to identify just those individuals to then go back and try lithium again.
A clinical question. You're talking about mapping your whole genome. How do I do it?
How do you do it? Well, from a research perspective, it would be something you could. How much?
1,500.
Yeah.
Where do I go?
If you wanted to do it from a research perspective, you would, for your own personal interest.
I do it for myself.
For yourself, you can Google it. You can find companies that will do it for you. It's a lot more expensive than $2,000 is the research price. And so the patients that come through the Macquarie Clinic are consented to research, and their genomes are sequenced on a research basis. But that data is not returned to the patient because it's done on what we call a de-identified way. Unless something critical comes, we find something critical that's relevant to your health, then we have an ethical obligation to let you know about that. But if you wanted to go and do it as a private individual who is interested in doing sequencing his genome and perhaps want to see whether you carry risk variants, yeah, there are companies that will charge you substantial sums of money to go and do that. What is the utility of that? What's the use of that? Is very open, because it might tell you you're at risk of some stage in your life. You might have a 20% increased risk of diabetes, or what do you do with that information is a bit unclear.
Conversely, it could put you on the front foot for a clinical trial.
Oh, I see. Yeah, well, in terms of determining your genotyping for a clinical trial, I would say a clinical trial will have very specific criteria that they will test. And it may not be a genetic test. It might be a biochemical test, a blood test. It might be an imaging test, where you do an MRI, for example. There are different criteria. The genetics is just one of those. So each clinical trial will try and achieve that personalised medicine approach using different means.
One of the workings with the genome, genetic factors, but one of the significant environmental factors that they found.
Good question. What are some of the significant environmental factors that might trigger the disease? This is really open to a lot of conjecture. And I showed that 6-step process, and we do believe that it's a combination of environmental and genetic risk. And the environmental exposure, and there have been studies that suggested particular agricultural chemicals, or smoking, or excessive exercise. We see some elite athletes appear to have an increased incidence. But it's difficult to say that across the board, because you might need the genetic risk while you might be exposed. For example, if you're a smoker, and we saw with the centenarians, if you don't carry the genetic risk, then you don't respond in a negative way to the same extent from the exposure to the so they've managed to escape the negative impacts of the environmental exposure. So it's difficult to know without knowing if you carry that susceptibility, without susceptibility to a particular environmental environmental exposure. Without knowing the genetics, without being able to stratify, you can't tell which of the critical environmental impact risk factors. But some of those I mentioned have been speculated. Now, smoking is an unusual one, because smoking was historically been thought of as a strong negative environmental risk factor. But there have been studies that suggested smoking is protective. And we saw that in Rebecca's talk, where she mentioned that high cholesterol is actually considered protective. So how do you tease these things out? It will come.
I have a question.
Talking about the genetic links in a family, if one of the siblings has ITP, which is a long name like idiopathic thermolytic purple, I can't pronounce it. It's a very long name, but ITP, which is you have excessive bleeding and it has to do with your platelets, and the other sibling has MND. So is there some kind of link in that?
No, I think you're talking about two quite distinct disorders. You wouldn't test one as a risk factor for the other in that case. No.
So it doesn't have it.
No. Shoot.
Is that me? Hi, Ian, sorry for working in late. Before you showed a slide about the 6-step hypothesis and the number of steps that it took someone to come across to develop MND seemed to change based on the gene that they might have had.
Yes.
So can you explain why this is the case?
Right. So this is what we talk about the effect of the risk variant. The risk variant we may inherit might be very weak. It might be moderate or it might be strong. Now, you can imagine if it's a weak risk, then you need many. And you can see that in the general population where there's heart disease or diabetes or some of these. Obviously they're caused by, or likely to be caused by, many different variants. Now, at the other end of the spectrum, there are genetic variants that confer a very strong effect. And that's where these ones that carry what we start to use the word mutation. And SOD1 is one that's very strong, and it can take multiple steps because it triggers multiple biological cascades in your body, and each one of those changes in your body will be they believe it's 5 steps. Now, you need a 6th step, and not everyone in that family will get that 6th step. And that's why we see some individuals who've inherited the mutation never develop the disease, because they missed out on that 6th step. Other mutations only take 4 of the steps, so you need another 2. And so you see that. You see some of the families will have less of an incidence within the family. So while individuals are inheriting the mutation, not everyone is inheriting the disease, is developing the disease. So you need these additional steps to trigger, to reach that threshold.
So that might explain twins then, who have the same genetic makeup but might develop MND at different times? Is that right?
Yeah, good question. So it could explain twins, identical twins, particularly if they inherit a mutation but don't develop the disease at the same time. Or twin studies are fantastic from a genetic perspective, and we have a particular focus on recruiting identical twins and non-identical twins as well, because we generally consider they have had the same environmental exposure at least until they've gone their separate ways. So, for example, they might have lived in the same family environment for the first, say, 18 years of their life. And they've had the same environmental exposures. So we say they're controlled for that environmental exposure. What are the differences? And often those differences can be largely genetic. And they are very powerful to tease out there. And so we have a collection of identical twins that we've been recruiting for years.
So just an add-on to this gentleman's question about the environment. Is there anything that suggests that a virus can bring on this?
It could well be, yes. So any of these sorts of exposure to, were at this stage unknown environmental exposures, and a virus is one that, yes, you would be exposed to through the environment. These environmental exposures could be anywhere from your exposure during the womb to your exposure throughout life. And the sixth step, the multi-step process, goes some way to explaining why it's a late onset disease, because you've accumulated these over your lifetime. And some accumulate them faster than others, because we know that the onset of MND can vary widely from those in their 20s, 30s, to those who are in their 80s and 90s who make onset of the disease.
Just to add to that, I'm wondering, when we talk about environmental triggers, we were talking about the influence that it could have on the actual expression of the gene.
Oh, absolutely, yeah, good question.
So that would include not only a viral infection. I mean, Tecfidera was originally for retrovirus, right? So we're looking at that as well as the exposure to environmental factors like heavy metals. So would you say that the combination of those two would compound in the organism?
Yeah, so the question was around multiple environmental exposures. Absolutely. So your exposure to an environmental, how does an environmental exposure translate years later to a step leading to that threshold? Well, it turns out that environmental exposures actually do modify our gene expression. It's what we call epigenetics, and it's widely accepted. This is widely known that even something like smoking changes the way your genes are expressed, and the way those genes are expressed may translate years down the track. And it's come about through how while it might not be a change in your genetic code, your genes have been, the expression of your genes has been changed by your exposure to an environmental factor. So that's the mechanism through which you have the combination of the genetic and the environmental risk interacting.
Sorry, Ian, not so much a question, but just a comment. So I'm also a researcher, and I think Ian and I both get questions all the time about the genetic susceptibility to disease, but also environmental triggers. And the environmental triggers and the questions around environmental triggers comes up time and time and time again. And there's a research group in Brisbane, and what they've been doing is developing an online questionnaire about triggers that someone might come across in their lifetime from a young age up until current age. So this is around occupation, where have you lived, have you been struck by lightning, or anything like that. And so once this questionnaire is validated, we're hoping that it will go live so that we can start collecting this information. Because we need to collect a large amount of information about environmental triggers before we can actually determine whether or not something is a trigger for the disease rather than something that's happening by chance. And I think the group in the Netherlands that's leading Project Mind, the big genetics initiative that Ian's been talking about, they've done a lot of historical research on environmental triggers. They're the ones who have come up with the idea of smoking and physical activity as being potential risk factors. So we're hoping that from the Australian side we can also contribute that information to a one-body initiative.
I like that. The being struck by lightning. Anecdotally, being struck by lightning, we do have what I would think to be an unusual number of cases in our research cohort that have been struck by lightning. Yeah.
Or people who are electricians?
I don't know, maybe they're... who knows?
Is there a way that people who don't have MND can contribute to the research? Obviously, apart from the obvious way of donating money, is there any other way that people who don't have MND can assist?
Well, from our genetic studies, we're definitely always looking for control individuals. But a control individual is somebody who is generally unrelated to an MND patient, so that they're unlikely to have inherited a gene. But we do like particularly to be what we say age matched as well. Cases to be a similar age to MND patients, and absolutely, and they're almost the hardest to find. We can get patients eager to participate in research when they come through the door. Sometimes we take blood, that's the simplest thing. Sometimes we take chunks of skin too. That's not too attractive for some people just off the street. But yeah, absolutely, controls is something we're always trying to control individuals, always trying to recruit.
Can I just add to your answer to this lady's question. Professor Rowe does have a system like that. Those without, I contribute either blood or my hair. And I think he does that with the spouses
Right.
those at his clinic.
So each of the particular clinics that recruit MND patients for research will recruit control individuals. And yes, we do hair, blood, urine, sometimes skin. Yeah.
Great.
Okay.
Is that all the questions? Well, let's thank Professor Ian Blair. That was excellent. And, we have a little token of our appreciation.
Thank you.
Thank you for punching above your weight.
Oh, my pleasure.